Okay, I think we have most of our registrants joining our webinar right now, so we will get started. So good afternoon, everyone, and thank you once again for joining us today. My name is Cassie, and on behalf of GenScript, I would like to welcome you to this exciting webinar on a novel synthetic self-amplifying RNA system and LNP delivery. The approval of mRNA-based COVID-19 vaccines marked a groundbreaking moment in modern medicine, proving the immense potential of mRNA as a transformative therapeutic modality. However, while traditional linear mRNA has revolutionized vaccine development, the short half-life presents a challenging and requiring high dosage to achieve a sustained therapeutic effect. So this is where self-amplifying RNA emerges as a game changer. By encoding a viral replicase along with the target protein, self-amplifying RNA enables continuous protein production, requiring significantly lower dosages while achieving prolonged efficacy. And this makes self-amplifying RNA a promising next-generation approach for RNA therapeutics and vaccines. So in today's webinar, we will explore how GenScript developed a novel self-amplifying RNA system to enhance therapeutic applications, the potential of an alpha virus-based self-amplifying RNA platform as a powerful and durable tool for vaccines and therapeutics, and lastly, a comprehensive one-stop solution for self-amplifying development, including sequence design, synthesis, purification, and LNP encapsulation. And we were happy to have Dr. Kevin Chen with us today, who will be sharing his valuable insights into this advancement. A short introduction for Dr. Kevin Chen. He's the head of the mRNA and plasmid department at GenScript Biotech. He holds a PhD in biology and microbiology from South Dakota State University, where he studied molecular microbiology. And since 2022, he has led the mRNA and plasmid teams specializing in plasmid preparation, IVT, mRNA synthesis, and LNP delivery. On top of that, Dr. Chen has authored more than 15 peer-reviewed papers, including a 2023 Nature publication on genetic code swapping to block viral infections. So in this webinar, we encourage you to engage, ask questions. You can see that there's a Q&A chat box over there. So if you have any questions, please key in your questions over there, and we will answer them one by one after Dr. Kevin Chen's presentation in this webinar today. So without further ado, we'll get started, and please join me in welcoming Dr. Kevin Chen. Dr. Kevin, you can start.
Okay, thank you, Casey. Can you hear me?
Yes, I can hear you clearly.
Okay, yeah, thank you all to attending this webinar. So today I'm going to show some progress in my lab about the self-amplifying RNA and the delivery system. So my talk will mainly cover these four different aspects. The first one, I'm going to do a little bit background introduction, and then I will introduce the self-amplifying RNA and lipid nanoparticle platform developed by GenScript, and then I will show some case studies using our saRNA vectors. And in the last part, I will introduce our servers. So as we know that MI therapeutic has been a hotspot in recent years, and many different drugs has been developed, especially for the COVID-19 MI vaccines. So there's multiple applications for the MI-based technology, such as protein replacement, and of course the MI vaccines, and also genome editing using the Cas9 mRNA. So, and right now there's multiple
Hi, Dr. Kevin
commercialized.
Chen.
Yep.
So sorry to interrupt you over there, but we have multiple feedback saying that the background noise is quite loud. Is there any way that you are able to move to somewhere else?
Let me try.
Because there's someone speaking at the background, and it's quite they can't hear your presentation clearly.
I just worry about the internet. So,
yeah, that's true.
let me find a good place.
Yes, thank you.
This one, yeah, this one's okay.
I think right now it's okay. I don't hear any background sound anymore.
Okay, so yep. So as you can see on the list on the right side, there's multiple commercialized mRNA vaccines. The COVID-19 mRNA vaccine developed by Moderna and also biotech, and there's some other type of vaccine like the Volvax and Abogene's. They develop a COVID-19 vaccine, but it's mainly in some Asian countries and other type of vaccines. And especially for the last one, I think I have a more detailed slide here. So you can see in last year, actually 2023, Japan approved saRNA-based COVID-19 vaccines, and which was developed by CSL and Actaris therapeutic companies. And here, this slide, I just did some research and summarize all the peptides that use the saRNA vector technology. You can see there's multiple like target the SARS-CoV-19 protein and inflow enzymes, and also I think there's some HIPs and neoantigens cancer vaccines. So the many big pharma company has put tremendous resource on the saRNA peptide development. So so now I'm going to introduce a little bit about the self-amplifying mechanism. So in this slide, you can see this is the traditional linear mRNA. So basically, you have mRNA sequence, and then the coding sequence translated into a target protein. And but for the self-amplifying RNA, basically there's replicase genes, which was encoded by the non-structured protein 1, 2, 3, 4, and in the downstream there is a subgenomic promoter that can replicate the target gene here. So after the self-amplifying RNA produce the replicase, it will recognize the subgenomic promoter here and replicate the GOI for multiple copies. So eventually you will see there's a multiple copy of a GOI, and will also produce multiple copy of target proteins. So that's the whole idea for the self-amplifying RNA. And so in the second part, I'm going to introduce our saRNA and lipid nanoparticle delivery systems. So at the very beginning, we used the traditional VEV virus-based self-amplifying RNA vector, but later we found that there's multiple issues like lower expression levels, and also it's a long targeting they don't have a cell-specific expression patterns. So our idea is like, can we find some novel saRNA vectors from the alpha virus family and see they can have a cell-specific type of expression, and at the meantime, and we increase the protein expression level significantly. So that's the trick question. We want to develop our own saRNA vector at the very beginning. So follow this idea, we synthesize multiple saRNA vector from the alpha virus family, and then we do a in vitro cell assay to screen any higher level saRNA vectors. And after we have the first round of screening, we test this vector in our mouse animal models and try to characterize their expression patterns. So eventually we did like a screening to 30 different alpha virus sequence, and our in vitro and in vivo data indicate there's some saRNA vector shows better protein expression, and also cell-specific expression patterns. So, and we choose the top 3 saRNA vector in our downstream test. So first, we try to compare the traditional lipofectamine-based transfection method, and also the lipid nanoparticle-based delivery system. So you can see on the top is the lipo-based transfection, and on the bottom you can see the LNP-based transfection. We use the EGFP fluorescence reporter genes here, and on the right side you can see the quantification data. So basically, the idea is like the lipo nanoparticle delivery system can significantly increase the transfection efficiency. And then we try to, because as we know, there's multiple modification to the linear mRNA, like the pseudouridine or the F1 mass pseudouridine, etc. So our question is like, is there any modification required for the self-amplifying RNA? So follow this idea, we designed a different sequence. So the unmodified saRNA vector sequence, and M5C modified sequence, and 5M1U modified sequence, and pseudo-U and 1 pseudo-U, and also combine the M5C and 1 pseudo-U here. And you can see we test this sequence in 2 different type of cell lines, the HEC293 cell and also the A549 cells. A549 cell is more sensitive to the stimulus. So, and you can see the M5C data indicate much higher protein expression, probably a little bit longer protein expression too. We conclude that the M5C indeed help the self-amplifying to increase the protein expression. And here is like a beta test, like we work with Dr. Luo in the Nanyang Technological University in Singapore, and we make this modified saRNA vector and sent to his lab, and he did had a comparison. You can see the modified version showed much higher protein expression than the unmodified version. So, and then the next question is like, what about the poly A tails? Because as we know that the linear mRNA, they requires a poly A tails, right? And typically we design like 100A or a little bit longer, like 120As to have a good protein expression. So, and on the left panel, you can see the different lengths of the poly A tails designed in the linear mRNA. So that's pretty much is expected, because we know linear mRNA requires a poly A tails, and the optimized length is about 100 As. And then we try to characterize the poly A tails in the self-amplifying too, and we design like a different lengths, 40 As, 70 As, and 100 As. But surprisingly, we did not see the poly A tails affect the protein expression in the self-amplifying RNA, and we later understand, because in nature the alpha virus, they don't have a norm poly A tails in the native sequence. So in our design, by default, we just design 40 As for the self-amplifying RNA sequence. So after we characterize the poly A tails, the modification, and the delivery system, we also want to know the dosing, because as I mentioned in the previous slides, the self-amplifying RNA can replicate itself, right? So, which in other words is it can lower the dosing to have the same level of protein expression compared with the linear mRNA. So we use the lipofectamine transfection method, and also the lipid nanoparticle delivery method, and tested this design in 3 different type of cells: the HEC293 cells, and the A549, and the Jerker immune cells. And as you can see, we have 3 different saRNA vectors, and also we have a linear mRNA sequence as a control. And meantime, we have a saRNA as a control too. So so in the LNP groups, the linear mRNA, we load 100 nanograms, and the same thing for the saRNA. And we load only 1/10 of the self-amplifying RNA. And you can see in the 3 different type of cell lines, this 1/10 dosaging also reach a similar level of protein expression as the linear mRNA and the saRNA. And in Jerker cell, it even shows much higher protein expression than the linear mRNA and the saRNA. So after we characterize the dosing for the self-amplifying RNA, we also want to know how about the immunogenicity. So to find the answer to these questions, we also designed this experiment. Again, we use the same set of sequence, the linear mRNA and the saRNA RNA as a controls, and 3 different type of self-amplifying RNA with only 1/10 of loading here. And you can see the control is like without any MI samples, so it has a quite lower immunogenicity. And among these 3 different type of self-amplifying vectors, they shows different level of immunogenicity for the IL-6 and interferon alpha and the IP10s. And for example, the self-amplifying RNA-4, it has a little bit higher immunogenicity than other sequence. And the number 2, the SA number 1 and number 2 shows much lower compared with the number 3, number 4. I'm sorry, it's a number 1 and number 3 compared with number 4. And we also characterize the MI abundance, like so here is the 100 nanograms linear mRNA, and the second is 100 nanograms saRNA. And here we only picked 1 self-amplifying RNA and load 100 nanogram too. So you can see that the self-amplifying RNA has much higher accumulate RNA in the cells, which indicated that it replicated itself. So after we characterize all these questions in the in vitro assays, and then we want to know what's going on in the in vivo animal study. So as I mentioned, we picked 3 saRNA vector, -1, -3, and -4. And we picked the linear mRNA as a control here, and this is a negative control. So you can see this is the timeline: day 1, day 2, day 3, day 6, day 10, and 2 weeks later, 3 weeks later. You can see the luciferase. After 1 week later, the luciferase was barely detected in the group injected with the linear mRNA. But for the self-amplifying RNA groups, these 3 groups, like after 3 weeks later, we can still detect some expression of the luciferase in the mouse. So we also checked the body weight change for all these groups, and they barely see big difference. They're almost the same. And then we we quantify the fluorescence expression data signal from these animals, and you can see the linear has a very strong expression at day 1 and day 2. That is expected because linear use a cap-dependent translation mechanism to turn on to express the protein. And the self-amplifying RNA, they need to replicate itself, and then they produce the target protein. So they have a different efficiency at the very beginning, but after like a couple of days later, especially after 1 week later, so we can still observe quite a strong luciferase expression in the mouse with saRNA sequence or samples. But for the linear MI, they barely gone. So, and as I mentioned at the very beginning, so we want to answer 2 questions. The first question is that we want to find better or higher protein expression saRNA vectors. And the second question is like, because we know virus, they have different tastes. They prefer to target different cell types. So we want to see is there any cell-specific targeting in these SNR vectors. So we try to see the bio distribution of this SNRNA proteins expression. So again, we use the luciferase reporter gene here and check the expression in different organs like the lymph nodes, the brain, the kidney, the lung, the spleen, and the liver, also the heart. And you can see this group is the linear MI, and as we expected, it's mainly expressed in the liver. About more than 90% is expressed in the liver. But for the self-amplifying RNA, -1, they have prefer like in the kidney, in the spleen, and in the heart. And for SNR-3, it's preferred in the spleen. And for the SN-4, it mainly distributed in the spleen. So that was really excited when we see this data. And then we try to see what happened to the lipid nanoparticle delivery. So because we use the same formula for the LNP here. So I didn't show the data here, but later we found we use a fluorescence dye to label the LNP, and we found that the LNP is, for all these samples, they are still enriched in the liver, which means that the LNP delivery is the same for all these different RNA sequence. But because of the mechanism, the self-amplifying RNA shows different biodistribution here. So in the third part, I'm going to show some case study here. So as you can see, in GenScript, we also have an antibody department, and we kind of have some project work together for a long time. And one day, like one of our colleagues complained to me, it's really challenging to get antibody to the transmembrane proteins like the GPCR or whatever. So, and then we have the idea, why not try the saRNA method to express the transmembrane protein and to immunize the mouse and see whether or not we can get the antibody we want. So we picked one transmembrane protein, which is called CCR9 from the humans. It's a 7 transmembrane protein, and they have failed in to get this antibody using the protein or plasmid DNA-based immunization method. So, and we clone this gene into our self-amplifying RNA vector, and then make the LNP and inject into the rabbit. Here, for this case, we use the rabbit. And for the lipid nanoparticle formulation, we use the typical SM102 formulation here. And as you can see, we use the FACS to screen the serotonin, and you can see that after the second immunizations, we detect positive signal from the serines, and these are the negative controls. And then we also eventually we overexpress a few clones in mammalian cells and get this antibody and try to characterize their binding efficiency. And so in summary, like we use the FACS to screen about 400 positive clones, and we use the NGS sequencing to pick the top 20 clones, and then we validated these 10 recombinant clones, and 9 of them shows very good FACS EC15 result here. And then the second case is about the Claudin 18.2, and as we know that Claudin 80.2 is a very hot target. It has been well known that it's related in the stomach cancer development. So again, here we try to compare the difference of using 3 mRNA formats: the linear mRNA and the self-amplifying RNA, and also the circular RNA. And again, we use the SM102 formulations, and but in this case, we use the mouse for the test. And we use the CHO-K1 stable cell line overexpressing the antigen Claudin 18.2 in our screening test. And you can see after the cell fusions, the linear mRNA shows a little bit higher titer in the serines, and then the circular RNA shows a little bit low, and then the self-amplifying RNA shows even low titers here. But after we pick the clones and continue the immunizations, like after the third immunizations, and we check the titers in the serines, you can see the linear mRNA has about hundreds here, and the self-amplifying RNA has average about 200, which is like double higher. And the circular RNA is about 120 or 130 here, average. So that's quite interesting, because the self-amplifying RNA shows much higher, and in our previous data, it can last much longer protein expression patterns in vivo animal study. So the last case is we work with some groups, and we try to develop a COVID-19 vaccine using our self-amplifying RNA technology. So we choose the RBD antigen here, and but this project was held eventually, because the partner they just they don't have more funding to continue this project. But we checked the serine titles in the samples, and you can see even after 3 months later, the self-amplifying RNA produce quite a high antibody in the serines. So in the last part, I'm going to introduce the self-amplifying service package. As you can see that we have different scales, like micrograms to milligrams, and then we can provide the cap options because in nature the alpha virus they use a cap AU, which means as the first codons in their sequence. So usually by default we provide a cap A. And then for the purifications, we use the like the HPLC or the regular lithium chloride to purify the saRNA vectors sequence. And we provide different QC tests to characterize the quantity of the saRNA. And as I mentioned in the previous slide, that we developed 3 different types of saRNA vectors, -1, -3, and -4, and they show different characterizations. They have different biodistributions too. So it can be used for different applications. And for the lipid nanoparticle servers, we provide like different scales, and also it can be used in IM or IV, different administration route. And for the lipid nanoparticle, we also provide different ionizable lipid, the MC3, which is used for the saRNA, and the SM102 and ALC0315 that's the typical ionizable lipid used in by Moderna and biotech. And the MP01, we use this lipid for genome editing, and we found quite a good genome editing efficiency. And the QC test, the encapsulation efficiency, the PDI, and the data potential, etc. So here is just a slide to show our patent that we have been submitted and also granted. So we have like the GenCap patents, which is a very, it's kind of like a alternative option for the CleanGap provided by Trinec. So this is the details patents approved. And there's some difference I showed here. So the main difference is that we use different templates and to add the Cap1 structure to the RNA sequence. And we also have some patterns for the circular RNA and the way to make the circular RNA, and also how to produce scarless circular RNA, etc. And as we know that GenScript also have some GMP facilities. So when our customer finish the R&D works and they want to move the work to IND or clinical phase, so we will use our GMP facility and capability to provide this service to our customers. So for the plasmid DNA, we have grade C and level C and level A clean room, and we can provide different scales like 10 liter, 100 liter, and 200 liter fermentation and bioreactors. And for the mRNA facility, we also have the level C and the level A clean lab, and it can be used for PD and for GMP level sample productions. So our GMP grade MI capabilities can cover different phase, like the IND phase and phase 1, phase 2, and phase 3, and even after the BLA, we can provide a full GMP service to support the projects. So yeah, that's pretty much all the slide that I want to share with you today. And if you have any questions, I'll be happy to answer. Hey, Casey.
All right, thank you, Dr. Kevin Chen. Wow, we have quite a lot of questions from the audience, so let's just go through it one by one. So the first question, have you experienced much frame shift with self-amplifying RNA?
Well, that's a good question. We have not checked yet because as I mentioned in my presentation, the first question, the first two questions we want to answer is like higher protein expression and also cell specific expression. So, but definitely we can check the frame shift and in our experiment. So the answer is not yet.
Okay, Ken. Let's move on to the next question. Can you have multiple promoters for multi-antigen mRNA, or does it have to be a single promoter with multiple antigens in one ORF separated by linkers? I'm thinking about how the replicase can distinguish between two self-amplifying RNA in a single formulation to have OTH replicated relatively the same.
If my understanding is correct, like, so you mean you want to put two different saRNA sequence in one lipid nanoparticle and deliver to the cells or tissues, and then see the replicase can distinguish the different promoters. Well, usually we don't do that way. Like for example, if we want to express two ORF or two genes in using the self-amplifying RNA, we will use the IRES or the P2A or T2A, like linker sequence, but it will be cleaved by the protease in the cells. In that way, we can like express multiple protein, and yep, that's the typical way we do. Yep.
Okay, thanks, Dr. Kevin. Next question. Do you think it has a higher immunogenicity as 10 nanogram of self-amplifying RNA is giving the same immunogenicity as 100 nanogram of linear and circular mRNA? If we use a higher amount of self-amplifying RNA for in vivo, then there will be implications.
Wow, that's a good question. Yeah, we did the test like um as we know, the self-amplifying RNA sequence is from the alpha virus. It's a viral sequence, so you can imagine that it can cause immunogenicity issues in cell or in vivo. So, and if we load 100 nanograms, we will see quite high immunogenicity. That's the reason we want to lower the dosing, so it can lower the immunogenicity, but at the same time maintain a good level of protein expression. And if we load higher amount of saRNA, it really depends on your downstream applications. For example, if you want to use for COVID-19 vaccines, it's okay to load a little more because you want to stimulate the immune system, right? So the stronger, the better. But if you want to use the self-amplifying to do the protein replacement or genome editing, you have to be very careful. Yep.
Okay, thanks, Dr. Kevin. Next question. In addition to EGFP, any other transgenes were expressed using the saRNA?
Yeah, yeah. We have like extract more than hundreds of genes for our customers, and we always keeping receive good positive feedback from them. And as I introduced in my slides, we tried a different transmembrane protein in using our saRNA vectors. So yeah, it has been quite a well validated. So yeah.
Okay, thanks, Dr. Kevin. Next question. How long was the saRNA stable for before it is cleared and expression stops?
That's a good question. So it's based on our data using the reporter luciferase. So you can see that the luciferase can be maintained for about 3 months. But I guess eventually it will disappear. So right now I can see like 3 months is still there, but the abundance I think is much lower compared with the beginning. So yeah.
Okay, thanks, Dr. Kevin. Next question, is there any DIR distribution data in vivo?
Yes, the DIR distribution data is in vivo. So what we did is we labeled the lipid nanoparticle using the DIR dye, and then inject this labeled LNP into the mouse, and then after like a couple of weeks or some days later, we kill the mouse and we check the biodistribution for different organs. And what we observe is that the majority of the fluorescent dye was detected in the liver, which indicated that the LNP did not affect the protein expression level or patterns for the saRNA, but the saRNA sequence itself shows or caused different biodistribution expression patterns. Yep.
Okay, thanks, Dr. Kevin. Next question, what kinds of factor makes the difference of biodistribution in vivo? Did you use the same LNP?
Yeah, as I mentioned in the slide, we used the same LNP, and probably I just explained in the previous question. So, our finding indicate that the LNP, because we use the same formulations, so the LNP does not cause the biodistribution difference for this saRNA vectors, but because they produce different type of replicase, and this replicase has different capability to replicate itself or skip the immune attack from the host cells. So we don't understand the mechanism, but the difference was caused by the replicase, because we choose different replicase here. So yeah.
Okay, let's move on to the next question. Is saRNA 3 the one offered as a service, or it is the VEEV saRNA 1, the replicon offered to researchers? We have used the service before, but how do we know which saRNA we have received?
Well, that's a good question. So typically, when our customer wants to order the service from us, they will like sign a agreement that they will use for research used only kind of things, and we will deliver the SN sample to our customers, and if later they want to move the project into like IND phase or phase 1 or clinical phase, we can discuss the license to our customer again. So and there's like a license fee to get a details information for this different saRNA, and yeah.
All right, thanks, Dr. Kevin. Next one, regarding to the use of mRNA LNP, even with some modifications, most of the RNA will still be delivered to the liver. However, the chart that you just showed demonstrated that the saRNA has a tissue distribution that is vastly different from mRNA. So what are the mechanism leads to these results?
That's a great question. So actually, every time I present my data to people, and they always want to know what the mechanism caused this different biodistributions. So honest to say, as I explained to other questions, and we don't know the details mechanism at this moment. Our hypothesis is that because this snRNA -1, -3, -4, they are from the different type of alpha virus species, and they have different type of replicase, which indicate they have different capability or to replicate in cell, and also probably different capability to skip the attack from the host cells. So that's our hypothesis, but we're still like doing experiment to validate this hypothesis. So maybe in the future we will show more data. Yeah.
Right, thanks, Dr. Kevin. Next question, do you offer saRNA GMP grade for phase 1 clinical trials?
Yes, the answer is yes. As I'm showing in my last slide, that we have the GMP capabilities, which was provided by our subgroups, which called ProBio. Um, um, So they mainly focus on the GMP grade mRNA and lipid nanoparticle productions. So they definitely can support your phase 1, phase 2, phase 3, even BLA projects.
Okay, next question. If most of the saRNA is delivered to the liver in LNPs, how is the saRNA able to express in other organs given that the saRNA should not be able to be released from the transfected cells?
That's pretty much the same question to the mechanism questions. So as I said, they have different capability to replicate in the cells. For example, or like the SN9-1, they don't have capability to replicate in lung cells, but the -3 they have a good replicate capability in the lung cells. Even though you have only 1 or 10 copy of siRNA, they can replicate it itself, right? So they will like go to 100 or 1,000 or even 10,000 later. And then after they reach the peak, the cell will, degrade all these RNAs, and eventually they will gone. But in this process, it will cause the different biodistributions.
All right,
Thank
thanks,
you.
Dr. Kevin. Yeah. Next question. The subsequent copies of the modified saRNA replicated in cells will not have the M5C modifications unless added by the whole cells. Does the effect of M5C only works in the first round of transfection, or how does it improve expression in subsequent rounds after replication?
That's a good question. So yeah, I agree with you. Like for the first round, that the M5C was in the template, and then after the second round of replication, the new generated snRNA sequence does not have the modifications. So our hypothesis is that the M5C can slow down the degradation of the template, and eventually will can amplify the final protein expression level. So that's our hypothesis, and I think it's quite possible. Yeah.
Okay. Thanks, Dr. Kevin. Next question. In your in vivo experiment, does the animal produce immune response against the viral backbone, the replicase? If yes, does it affect the vaccine efficacy?
Yeah, that's a good question. Yeah, definitely the animal will also um, produce the immune response against specifically the replicase. And actually, the replicate is a complex. It's made by non-structured protein 1, non-structured protein 2, 3, and 4, and they expressed like at different time points. So the animal definitely will produce immune response to this virus proteins. But as I mentioned, for vaccine, we don't focus on this issue too much because we want to stimulate the immune system, right? So you can think it as a kind of adjuvant in your immunization process. So yeah.
Okay, thanks, Dr. Kevin. Next question. Have you ever compared the saRNA, mRNA, and together with protein peptide in mice to see the antibody production?
Well, that's a good question. So I tried to do it, but it just I just didn't do it yet. So because right now we don't have a good projects to compare this way. So if there are any good targets that we need to compare the RNA and the protein or the peptide, then we can well conduct an experiment to see the difference.
All right, thank you, Dr. Kevin. Next one. How big is the construct with a simple EGFP? Do you think the uptake efficiency will differ with other transgenes?
That's a good question. So the EGFP usually about 1 kilobase pairs, 1 kb. So it's not a big size. And the biggest one we tried is spike protein from COVID-19, for COVID-19 vaccine development. And also another genes that we tried is big gene we tried is a Cas9, because Cas9 is about 3.5 plus the UTR, whatever. The other sequence is more than 4. It's about 4 kb. So yes, we tried to make some large genes in using our saRNA vectors, and so far because the functional assay was different. For example, the spike protein, we'll check the serum titers, right? But for the Cas9 protein, we will check the genome editing. So we have not checked the. I assume here is like the transfection efficiency. So we did not compare like the size of the gene during the transfection process. But I don't think the size will cause big issues here.
Okay, thanks, Dr. Kevin. Next question. Is it a self-amplifying RNA going to different organs with the same LNP?
Yeah. It's interesting. I was quite surprised at the very beginning too, like you guys. As I explained in other questions, so I think the replicase is the key to cause this difference. Yep.
Okay, thanks, Dr. Kevin. I think we have one last question before we end this webinar. So what's the maximum size of GOI?
I want to show my credit to Dr. Luo here, Dahai Luo. He's a professor that I showed the beta tester result in my slide, and I worked with him for some projects, and we have really good, experience together. And thank you, Dr. Luo. And yeah, sorry, Casey. What is the maximum size of the GOI? As in our hands, we tried like the maximum size about 5 kb. So totally like because the size of the saRNA is about 7, so 7 kb. So totally is about 12 to 13 kilobase pairs. So that's the maximum we tried, and so far it's good. The only issue that you have to worry about is like if you increase the length of the sequence, then you will have purity issues. So the longer sequence you have, the lower purity you might have when you generate this sequence siRNA samples. That's the issue that when we deal with the longer sequence. Other than that, everything is okay.
All right.
Thank you.
Yeah, I think we have completed all the questions. Good job, Dr. Kevin. So yeah, like I say, to everyone who are still on the line right now, this webinar is actually recorded, and I will send this the link to access the recording after this webinar. Okay. So as we come to the end of today's webinar, of course, I would like to extend a heartfelt thank you to all of you who joined us. And we hope that this session has provided valuable insights into the advancements of self-amplifying RNA and LNP delivery. And of course, a special thanks to you, Dr. Kevin, for sharing your expertise and shedding lights into the potential of self-amplifying RNA as an efficient and durable tool for medical application. So at GenScript, we are committed to driving innovations and providing comprehensive solutions for RNA research, from sequence design and RNA synthesis to LNP encapsulation. If you have any further questions, or if you would like to explore how we can support your research, please feel free to reach out to us via email. So once again, we appreciate your time and engagement today. So stay connected with us for future webinars and also updates on the latest advancements in RNA technology. So thank you, stay safe, and have a great day, everyone. Thank you and goodbye.
Thank you, goodbye.
Thank you, bye-bye.